will wait for another 5 minutes and then we'll begin the session. So we will start the session now. I'll share my screen. So is the presentation visible to you all? Can I get some response?
Yes, ma'am. Yes, ma'am.
Okay, okay, okay. Okay, so this is the session 3 for proteomics course, problem solving session. So it will cover this week 3 topics, 2D gel electrophoresis. So let's see a few questions, few problems, and I have also included few queries. A few of you had some queries, so we'll also look into that at later stage. This is the first question. Which of the following is not used for visualizing proteins? So these are basically different dyes which are used when you want to see the proteins after doing your SDS page or doing two-dimensional electrophoresis. So can I have some answer? Can someone answer this? It's a very simple. If you have gone through the videos, I think you will be able to answer this.
Ma'am, are you showing the questions? Are you presenting the questions?
Sorry?
Are you presenting the questions, ma'am?
Yes, yes, I'm presenting a question. The question one, which of the following is not used for visualizing proteins? You're not able to see this?
No, just there is just black screen popping up.
Is that a problem with everyone? Uh, Hello?
Yes, ma'am.
Yeah, are you able to see or it's
Yes, ma'am. The screen is not.
screen is not visible? Okay, I'll stop sharing and maybe do it again. Is it visible now?
Yes, ma'am, it is.
Yes, ma'am.
So this is the first question. Which of the following is not used for visualizing proteins? Kumasi blue stain, silver stain, ETBR stain, that is etheridium bromide, colloidal Kumasi G250 staining. So these are different staining dyes used for visualizing proteins. So you have to select which of the following is not actually a protein visualizing dye.
Is it ETBR, ma'am?
Correct. ETBR is not used for visualizing protein. It is used for DNA visualization. You're very correct. So coming to second question, while performing a gel-based proteomics experiment, proteins run in gel are generally stained using a particular dye. Why is this staining required? So we, as discussed in the previous slide, different kind of dyes are used for staining the protein. So what is the role of this dye? So let's see the options. The components of staining solution help the proteins to resolve properly. In order to visualize the proteins as spots or bands, or to see the quality of protein sample and pick protein bands or spot for mass spectrometry. Staining solution will remove any contaminant if present in a protein band. So, which of the following is the correct or are the correct options amongst the all?
I guess it's to visualize the proteins as bands. That is the
Yes, visualization definitely, as we discussed in the earlier slide also. So they are very important for visualizing the proteins. Else you will just see the your gel, and if you are not using any staining dye, you will not be able to see your proteins of interest. And any other options also? There is also one more option. So second is to see the quality of protein sample and pick protein bands or spot for mass spectrometry. So when you are doing a proteomics study, okay, so initially you perform a gel-based analysis. After that, now you want to identify a particular protein spot. Okay, so then you will use mass spectrometry for its identification. Okay, so once if you are using a staining dye or anything, you will be able to visualize, and then you will be able to use that particular spot or bend which you are seeing as a, maybe it's of your interest because it is changing in your test sample compared to a control sample. So it could be further used for even mass spectrometry based analysis, and you can also see the quality of a protein sample depending upon the uh, if you're using the staining dye. But if you're not using a staining dye, you will not know whether your protein has gone out of the gel or whether it is present, or what your protein of interest is even coming or not. Okay, so staining is a very important step in your gel based proteomics experiment. So coming to figure 3, so this is a 3D view of a, okay, so given below is, just a minute, someone wants to join. Okay. Okay, so given here is the 3D view of a region of a 6 2D gels. Okay, based on the figure provided, answer the following question. So these are six images of six samples you are having, six gel images. So what can be inferred from the given figure? So seeing, looking at the figure, you have to make some guesswork that what can be inferred. So we will see what are the options given to us. Option A is gel 1, 3, and 5. That is this, and this have similar profiles and may belong to one group of samples. Okay, so let's see gel 1, 3, and 5. So if you see the pattern, so even this is common in all the three. Even this region is quite similar. So you will see that I guess this is also present. This region is also present in this as well as this, and this is unique to this. But okay, so most of the regions are common in all the three. So we can think that it could belong to a one group of samples, meaning maybe they are in a group of control or test, whatever. So we don't know what is the profile, but we can just assume that they may be, just a minute. Okay. So it will be good if you all join right sharp at 3, or at least by 3 5 all of you join, because then every time I have to stop in between and uh, admit the person, so it will disturb all the other members also. So it's a request to all of you that at least join by 3:05 every time. Okay, so we will continue. So, this is the third question we are discussing for the people who have joined now. So, given below is the 3D view of six 2D gel images, and from these images, we have to infer certain things given in options 4. So we were discussing option 1 of A. Gel 1, 3, and 5 have similar profiles and may belong to one group of samples. So, looking at the images, currently we are thinking that this could be a possible option. Now we will look at other three options. Gel 1 and 6 are technical replicates of each other. So, technical replicates, anyone has an idea about what is technical replicate? Okay, so it is like you are performing the same kind of experiment. Everything is similar. You're taking the same sample. Let's say from a you are working on some bacterial culture. Okay, so you're using suppose you are taking that culture from a particular petri dish. Okay, and you are isolating it, and you want to perform proteomics experiment. Okay, so you are doing that experiment in replicate. Okay, so you are using two set of your appendix, everything, and you are using the same bacterial culture twice. You are keeping all the experimental procedure, timings, everything constant, same in both the things. So that is like technical replicate. So that means it's a technical replicate. So it should be ideally completely same because it is from the same sample. So let's see if that holds true in this. Gel 1 and 6 are technical replicate. So this is gel 1 and this is gel 6. So this region is common. Uh, so this, if we see this region and this green marked region, uh, this is quite different here. Even this region is quite different in this case. So I don't think this is a technical replicate because as I explained, technical replicates are from a same sample and they have gone through the same experimental procedure. So it should be ideally same. So this is false. This is completely wrong. So this option is gone. Now coming to option C. Gel 2, 4 and 6 may belong to the same group of sample. So that is 2, 4 and 6. So we have to see from this image what how similar they are. So if they belong to a same group, there will be some similarity in it. Okay. So as we discussed for 1, 3 and 5. So anyone wants to comment upon this? Do you think 2, 4 and 6, that is these three images, do they belong to the same group of samples? Uh, can you interpret seeing the gel image?
No, no. Same group of means similar kind of like organisms. That's your opinion.
So same group meaning, let's say you are trying to study two different groups. You're trying to study drug effect on some cell line, okay, and you want to see now proteomics analysis. So in a one set, you are providing just a minute. Someone wants to join. Okay. So same groups meaning you're doing an experiment. Oh, so sorry for the interruption. Uh, so what we were discussing, same group. So same group basically means let's say you are studying you are doing a proteomic analysis and you are you want to see a drug effect on a particular cell line or maybe an organism. Let's say we take a rat model. Okay, so you have given a drug to four rats and now you want to study it effect, and there is a four group of control. So there are four rats where you are not giving a drug treatment but just a placebo treatment, and now you want to compare their proteomic analysis. So the drug treatment group, that four rats, will belong to a same group. So are you are you understanding? So that one will be a one group, drug treated group, and another will be an another group. So basically they are different organism, but they belong to a similar group. So the more or less the condition, you are giving some treatment or you are trying to see like study a disease or something, so there will be a difference between the two groups. So
Now,
depending upon
can you
the,
give an example, like real life example?
sorry,
Can you give an real life example, like
for
same group?
same group. Okay, so let's say there is a study going on like diabetes. Okay, so just a measure of people, someone wants to study, are the diabetic people really obese? Okay, so what will they do? They will recruit diabetic people and also the people who are not having diabetes, non-diabetic group. Okay, and then they will calculate their body weight and height, BMI, they will calculate for both the groups. Okay, and then they will segregate both the groups. So one is a diabetic group, so more or less you expect that although the individuals are different, because of one common factor here, that is diabetes, or in case of other a drug a drug treatment or whatever, so they will have some similarity. Okay, so they belong to a one group and another is a control group. So they don't have a diabetes, so they belong to another set of class, another group. So because they all of them do not have the condition, do not have the disease, they all will have a similar kind of you can say characteristics. So here I am just giving you a simple example of BMI because it is very simple to understand. So you can say that you are monitoring 10 individuals and you found that more or less diabetes, they are having BMI value more than 23. And if you see in the control group, it is less than 23 for more or less. You can say that at least 70% population or 7 individuals from the 10 were having that kind of profile. Okay. So I hope you understand that same group.
Yes, ma'am.
Now, coming to option D, intensity for all the spots is same in gel 2, 4, and 3. This is 2, 4, and 3. So you can see 2, 4, 2, 4, and 3. So these three options. So this protrusion in a 2D, it gives an idea about intensity. So if you can see, it is not similar. Even in, you can visually see here it is different. This is different. This is also different. So this option is also not correct. So yeah, what about option C? What do you think? So this option is ruled out. B option is ruled out. A option is correct because we have gone through 1, 3, and 5 image and more or less they are similar. So what about C? Anyone wants to comment? Do they belong to a same group or a different group?
Option C may be correct. As you say, there should be
Hmm.
similar kind of organism, but there is like someone has affected by corona or that.
Exactly. Yeah, you're right. Option is also correct. So as I mentioned, same group will have a similar, it will not be identical. Whereas in case of technical replicate, because it is from the same sample or same individual, it will be completely, at least theoretically, it will be completely identical. So that is why if we compare the similarity wise, technical replicates will have highest similarity than if like from the same group, it will have lesser similarity than technical replicates, and from the two different groups definitely it will have a different it will have the least similarity. So coming to question 4, isoelectric focusing is an important step in two-dimensional gel electrophoresis. It is based on which of the following principle? option A is that the proteins migrate in an electric field till it reaches a point where pH is equal to its isoelectric point. Option B is that the mobility of the proteins in the electric field will not change with changing pH. That the proteins will move in the field until they reach their zwitterionic state. And option D is that the proteins in the electric field should not have any polar amino acid. So, what is the principle of isoelectric focusing?
Ma'am, it's option A.
Yes, next option, yeah. It's based
That
on
the
the
proteins
charge.
migrate in the in an electric field till it reaches a point where pH is equal to, correct, you are correct, where pH is equal to its isoelectric point. Please wait for a while. I'll just let people enter. So everyone, please join on time. It will also disturb other people. So we were discussing question 4 about isoelectric focusing. What is the principle? So option A is correct, but if you see option C, that the proteins will move in the field until they reach their zwitterionic state. So isoelectric point is a pH where it has both it has no net charge. Okay, so it will have a positive as well as negative entity. So that is called a zwitterionic state where the net charge is zero. So pH is equal to pi. So even here it is same. Both are meaning same, but the sentence is different. Okay, till it reaches a point where pH is equal to isoelectric point, and here that the proteins will move in the field until they reach their zwitterionic state, which means until they reach their isoelectric point. So it means the same thing. So even option C is correct here. So you should pay attention to this certain sentences and words. It might be tricky sometimes. So this is another interesting question. Okay, let me. Someone wants to join. So a polyacrylamide gel was casted and then a mixture of 5 different proteins was placed over the gel. Electrophoresis was performed at neutral pH. Okay. Given below is a schematic representation of gel before and after the voltage was applied. Given that the molecular weight for protein D is lower than protein C, whereas its isoelectric pi is higher among the given proteins. Okay, so what is the information given? Let's first discuss that. And then we have to find out which of the following is the correct match. So we, from this and this image, we have to figure out that which is the correct match. So what information do we have? So here for protein A, B, C, the pi isoelectric point is given 4.9, 4.7, 8.7, and 6.9. Okay. And another information is given is that electrophoresis was performed at neutral pH. Okay. So using this information, this is a very important information. So this will give you an idea about uh, like what will be the charge of protein, how it will migrate. Okay. So here pi value is given. So from that you can decide. And secondly, we are, so in the first case we are not applying voltage, and in the second case we are applying the voltage. So this is the sequence A, and uh, here we are applying the voltage. So here the charge of the protein will come into picture. Okay. So uh, you try to solve this, then we'll discuss. I'll give you some time. At least try using this information. Um, pH, an experiment is performed at the neutral pH. It is very important information. And secondly, you have been given all the PI values for protein A, B, C, and E. And for protein D, uh, the two information are given, but let's focus upon these four proteins first. So you try to first see from this information what you can infer. So I'll give you 5 minutes. Think about it.
And can I try?
Sure.
I guess it's option A.
Option A. Okay. That is A equals to 1. Okay. Any other one wants to try? We'll wait for a few more minutes. So can you comment why you think it is an option A? Do you want to give your reasoning behind that?
Yes, ma'am. Because first of all, if we see about the isoelectric point, the D has higher isoelectric point than all the other all the other amino acids. Um, D is more than or its acid more higher. Yes.
Uh,
So, ma'am, and and in all the four options, D is equal to 5. Okay. So higher isoelectric point means those acid, aspartic acid, these they will be
sorry,
like
sorry. You were not audible in the between. Your voice was breaking. What were you saying? Option D is what?
D. Like D has a higher isoelectric point than the other amino acids.
Correct, correct. You're correct.
Is so like higher isoelectric point means these are like some acidic amino acids like aspartic acid, glutamic acids. They have those high isoelectric points.
I think you are mistaking high isoelectric point. Aspartic acid, glutamic acid will have lower PI.
Oh, I mean,
You
it's
are
lysinurgene.
yeah, you are. I mean, you said opposite, I guess.
Yeah, yes, ma'am.
Yes, yes, yes. You are right. Lysine and all the basic will have higher higher isoelectric point, which is
Yes, ma'am.
correct. So, but why? so why, what does it has to do with D5? So you are saying that D is 5 because it has higher isoelectric point. So that is why it will be uh,
To go into the negative.
going towards the negative side. Okay, but have you also taken this information that the electrophoresis was performed at the neutral pH? So, what is the current pH and what is the pI of a particular protein? So, both has some relation uh, when it comes to what charge a protein will have. So, whenever you're
Excuse me, ma'am.
sorry,
Excuse me, ma'am. I think the answer is C.
okay, okay. So, can you explain that why it is C?
Okay, so since it was done in a neutral pH, and uh, the in the information given, it is said that we can notice that the protein E has a pI of about 6.9, that is near to neutral pH.
Correct,
So, if we look at the gel after the applied voltage, you can
correct,
see the tree has not moved anywhere,
correct.
which means that it is staying still and it is in its isoelectric state. So, I think the answer is E.
Yes,
And all the other is like 1 and 2 has low isoelectric point, so it has moved to the positive end, so it will be A and B looking at the values given in the question, and same with the C and D. And since D is higher isoelectric point, it will be further to the negative end. So 5 is D.
correct. So yeah, your explanation is correct. So someone has given option A. So I hope it is clear to you. So we'll also discuss about this. So one of the important information is, so you have pH was given, right, and PI was given. So this is the relation when it is so what the charge a protein will take, it will be dependent upon pH of the of your gel or electrophoresis, as well as the PI of your protein. So whenever your pH is greater than the PI, then the protein will have a negative charge. Okay. So I just forgot to this is for protein B, this is for protein C, and this is the value for protein D. Correct me if I'm wrong here. So if you see PI value is 4.9 for protein A. So here pH is 7, our pH is 7. So which is pH is greater than 4.9 value. So protein will have negative charge here. Similarly for protein B, even here pH is less than PI. So protein will have negative charge. For protein C, pH is so here pH is greater. So I have to correct this. pH is greater than PI. Sorry, pH is less than PI. 8.7 is given, which is less than PI. Array. Sorry. pH is 7 and your PI is 8.7. So your pH is less than PI. So your protein will take positive charge. And here it is almost equivalent. Okay. We are working at neutral pH. So it is almost same. So it will have no net charge. And similarly for protein D, the pi is highest of all. So definitely it will have a positive charge. Okay. So if we see uh, protein C and protein D, both of them will have a positive charge. Okay. So they will migrate towards the negative end when you are applying voltage. So here you can see in option C, D and D5 and C4 both will be here. And as we can see, in case of protein D, which is 6.9, sorry, this is protein E. Sorry for, I've forgotten to edit it properly. So protein E is having a same pH as pi. So it will not migrate as someone said. So it will be somewhere here. So protein E will be this 3 because pH is equal to pi. So it will not move. It will not have any charge. And in case of A and B, 4.9 and 4.7. So it will have negative charge. So it will move towards positive rod anode. Okay. So here will be your protein A and B. So option C is correct. So I hope this is clear to all of you. So pH and pi plays a role in what charge a particular protein will have. So whenever pH is greater than pi, it will have negative charge, and whenever pH is less than pi, it will have positive charge. And whenever pH is equal to pi, means no net charge, it is 0. So it will not move basically. So let's move to another question. Anyone still has any doubt? Because this was little bit tricky, I feel. Clear to all of you? Okay,
Yes, ma'am.
then. Okay, thank you. We can move ahead then. So this is question 8. So Rohit isolated human serum from dengue patients for doing proteomic analysis. He did depletion of the samples and performed 2DE for crude as well as depleted samples. Two of the gels obtained after performing the experiment are shown below. What could be the correct annotation of these gels if all the experimental conditions were constant for both the runs? Option A, gel 1 is for crude serum sample. Option B, gel 2 is for crude serum sample. C, gel 2 is for depleted serum sample. And option D is gel 1 is for depleted serum sample. So basically you have to identify of these two gel which is of the crude sample, crude serum sample, and which is of the depleted serum sample.
Ma'am, can you please
So
repeat?
I guess option A and C is the right one.
Okay. Option A is gel 1 is for crude serum sample, this one, and option C is gel 2 is for depleted. Yes, you are right. Gel 1 is for crude serum sample. So, I guess this I have also discussed in our live session, first live session, that whenever you are doing a protein experiment, proteomic study, first important step is you remove abundant protein, which are like albumin, globulins, which are present in very high amount, to see other low molecular proteins which are of your interest. So here, whenever you are uh, uh, doing your analysis with crude serum sample, definitely you will get a very big band of that abundant protein, and that is why you will not be able to see few proteins of low molecular weight. And this is of the second is of depleted serum sample. So this could be visualized from the two gel images. So you are correct. Option A and C are correct. So these are few queries that we have received. So one was in this video. So this video basically discussed about triazole method. So there was some technical issue where I guess the protein was uh, like shown in a band, like at the B, and the DNF was told to be in the um, at the debris at the end. So we are trying to fix that. Definitely, this is the correct way the proteins are separated in the by triazole method. You get first the bend of RNA, then the DNA, and protein is present in the lower uh, organic layer. So we are working upon this and trying to rectify it. Then there was another query regarding is gel in contact with the running buffer? Why running buffer is used? So here, l- just to give an overview, we can go to this video also, and then just to give you an idea. So the question is raised. Set the appropriate voltage and begin the run. All the proteins. So at 5:44 minutes, okay, so that was, so somewhere here, so here the multiple, here the apparatus for performing simultaneously uh, running multiple gels is shown. So you can see here multiple gels are loaded in a same apparator, and at 5:4, so at the end they are using buffer here. Running buffer is used. So the question is that is gel in contact with the running buffer? Of course, whenever you are doing any kind of electrophoresis experiment, running buffer is very important, because if running buffer is not there, you cannot uh, perform your I mean, the separation won't happen. Basic principle of any electrophoresis is the separation based upon charge and mass, molecular weight. So that is why they travel from one anode to cathode or cathode to anode. Okay. So in case of our proteins, if you see, we are using SDS, or we are using Kumasi blue dye. So they provide a charge to your protein. Okay. And that charge, on the basis of that charge, they are migrating. Okay. So now you require a current to flow. Only then they will migrate from one particular end to another particular end. So basically, your running buffer contains ions that conduct the electricity. So that is why running buffer is very important. So it is just not common to SDS page or electrophoresis for protein, but be it any kind of electrophoresis, running buffer is important, because without that, the current will not flow, and there will be no your proteins will not go from one end to another end. Okay. So I hope this is clear. Whoever has asked, I hope I have cleared your doubt.
Well, just a small doubt since
Uh-huh.
you mentioned this.
Hmm.
When the running buffer is in contact, like if we will there be any cross contamination between the proteins that is added into the well?
No, no, there will not be any cross contamination. I mean, cross contamination as in you are telling that it will react to your protein of interest, something like that.
No, ma'am. Will there be we will add different proteins to different wells, right? So when it is in
Oh,
the
okay, okay, okay. I got
running,
it. So you are telling that you are adding protein to different wells, and when you are using a buffer, will that one protein will go to another another well,
yeah,
something like
yeah,
that.
yeah,
So
yes,
it does not. It could happen definitely, but it does not happen. Why? Because whenever you are loading your sample protein in a well, along with your protein, you are using a loading buffer. So basically, in that there are certain chemicals you are adding which will give it a density, more density, so that your sample
yeah,
protein will settle in the well properly, so it will go down whenever you are loading, it will not come up. So that is
okay,
taken care, so that is why the contamination does not happen. So when you if you
okay,
forgot to add that loading buffer along with your protein, then this could happen, then this could definitely happen.
okay.
Mhm. So coming to question 3, so this at this we'll see the video just to give you a reference. Okay, so at this point somewhere, so I guess you are asking about 11:17. So I guess it is about this image. uh, as far as I understand, is the gel viewed under UV light. So basically, this video is discussing about when you are staining your sample with Kumasi brilliant blue dye. So, whenever you are using this stain, you need not to use UV light for visualization. You can use normal white light. So, generally, people use a white light pad because there could be few proteins, low abundant proteins, which will give a very light band. So, to have a very clear image, generally people are using white light band pad, like this white pad is used for visualization of uh, the gel image when you are using Kumasi brilliant blue dye as a staining dye. So the question was, is the gel viewed under UV light? So it was not used under UV light, it was used under normal white light. So this comes to the end of our session. But if you have any doubts or you want me to discuss any question that we have discussed in this current session, or even in any previous session, you can ask now. And those who had raised these queries, I hope it is clear now.
Excuse me, ma'am.
Yeah, please go
Yes,
ahead.
ma'am. In the assignment one, the number 10 question, can you please discuss that? There is a question about BLI experiment in the
Okay,
assignment one.
BLI experiment. Okay, I'll note this down. So maybe we can discuss this question in the next session. Okay, so I'll discuss this in the next session. So this assignment one date is gone now, the submission.
Yes, ma'am. Yes, ma'am.
Okay, so it is gone. Okay, then we can discuss that. So we will discuss this in next session because it will take a few more minutes. So we can continue in the next session. Okay.
Okay, ma'am.
The last question, right, in the assignment one,
Yes, ma'am.
BLI. Okay, okay,
This one, the last question, and in the week and assignment two, the first question. These two questions.
okay. Assignment two, first
First
question.
question.
So even assignment one, submission date is over now.
Yes, ma'am. Both the assignment dates are over. Both for
Okay,
assignment one and two.
okay, okay. Sure. Then we can discuss this. Definitely we'll discuss this. Okay, so in the next session we will discuss these two questions as well as we'll discuss few questions relating to week 4. Okay, week 4 content. So I'll stop
Ma'am,
my.
one small doubt.
Sure.
Uh, it's just regarding the assignments. I couldn't do the first two assignments due to some personal issues. So, is it a problem? Like, uh,
So I mean, there are certain deadlines. So I don't know actually that under what conditions they will consider it or not. So because the deadline is
okay.
already gone, right?
Yeah, yes, ma'am.
Yes, ma'am. Yes.
So basically, for the course completion, you need to complete the assignments.
Okay. Okay.
It
Okay, ma'am.
whenever you are I mean giving the exams, and if you want the certificate for that, the completion of the assignment on time is mandatory. So currently I don't think there is I mean any option available that you can submit it later on after the due date is over. But if you want, you can write to the mail to the NPTEL. There would be some support team available. So if they agree,
Okay.
then it is up to them.
Okay, ma'am. Okay. Thank you. Thank you.
Okay, no problem. Stop sharing now.
And ma'am, look,
Sure, yeah.
yes, ma'am. And if possible, please discuss another question, please. The week two assignment, question number 7.
Sorry, I was not able to hear you properly. Your voice was breaking.
Yeah, yes, ma'am. I was asking if possible, can you please explain another question from the previous assignments? Like it was from week two, question number 7.
Uh, week 2, question number 7.
Yes, ma'am.
Okay, so this is from this year's assignment, right?
This year, yes, ma'am. I don't catch you.
Okay, okay. So yeah, I'll note this. So in the next session, I'll discuss this. Question number 7 and question first. Okay, sure. Sure, we'll do that.
Thank you, ma'am.
Okay, thank you. Okay, bye bye. Have a happy weekend.
Thank you, ma'am.
Thank you, ma'am.